Welcome everyone to Journal Club 3. The purpose of these journal clubs is to emphasize published work that covers some of the analyses that we've discussed in the PAMCA Bioinformatics workshop. So in workshop 3, we covered population structure and different approaches to analyze population structure in the Anopheles gambiae 1000 genomes dataset, including analysis like PCA. And today, Alistair Miles will discuss the 2017 paper in Nature, Genetic Diversity of the African Malaria Vector Anopheles gambiae. Alistair is the Malaria Vector Surveillance Lead at the Wellcome Sanger Institute. And as we go along, please drop your questions in the chat box and we will answer them, some of them as we go and some of them at the end, but we should have plenty of time to answer questions. So take it away, Alistair.
Well, thank you so much, Brandy. Let me just share my screen and we will get started. Okay, so yes, as Brandy mentioned, the paper I thought I would talk about today is this paper, Genetic Diversity of the African Malaria Vector Anopheles gambiae, and this was the first paper that we published from the Anopheles gambiae 1000 Genomes project. And as Brandy mentioned, this paper was published in 2017, so we're going back in time a little bit here. And what we reported in this paper were the results of all of the analyses that we ran on phase 1, so the first phase of the AG1000G project. And because it was quite a big project, we broke it up into several different phases. And the first phase, we just looked at 765 mosquitoes, and most of those were Anopheles gambiae, hence the title focusing on Anopheles gambiae, but there are also some Anopheles coluzi in the data set as well. And I thought this would be a good paper to look at this moment, because as Brandy mentioned, the last training workshop focused on population structure, and a good part of this paper includes and covers analyses of population structure. So that's the main part of it that I will try and focus on today. But also in this paper, we ran a number of other analyses, which are things that we're planning to cover in further workshops in the data analysis training course. So I thought this would also be a good moment to introduce some of the analyses that we will be covering later. So anyway, hopefully it's a useful paper to cover now. So just to get started, let me talk about the mosquito specimens that were sampled and sequenced for this paper. So in AG1000G phase 1, there were mosquitoes from 8 countries. So the countries we sampled were Guinea-Bissau, Guinea, Burkina Faso in West Africa, then in Central Africa, Cameroon, Gabon, and Angola. And then in East Africa, there were mosquitoes from Uganda and coastal Kenya. So there was a total of 15 sites across 8 countries. When we made this map, we were looking for a good way to try and represent just a little bit of the kind of variations in the underlying ecology and geography, because obviously the range of these species is very broad and covers a whole range of different ecological settings. And there's major features like, for example, the equatorial rainforest, as well as the rift system in East Africa, which we're particularly interested in their effects on things like population structure. So, so this map here, the background layer, is actually from a separate piece of work, which was trying to classify different ecosystem types. So yeah, just to try and bring out that range of different underlying ecologies. So the only other thing to mention is that in addition to the 765 wild-caught mosquitoes that were analysed in this project phase, we did also have some crosses. And so these were crosses that were performed in the lab. And we had 4 crosses, each of which had up to 20 different offspring. And we sequenced those mainly as a way of quality control. So we used those data for quality control of the data that we then generated from the wild caught samples. So there's sort of 4 main parts to the analyses that I wanted to cover. And the first one is genome sequencing and our discovery of nucleotide variation, and then our analyses to summarize the nucleotide variation that we found. So by nucleotide variation here, I mean single nucleotide polymorphisms, single nucleotide differences between the mosquitoes that we sequenced. So all of the mosquitoes were sequenced using the Illumina HiSeq platform, and all mosquitoes were subjected to whole genome sequencing, and the target depth of coverage was 30x. So we had deep sequence data for individual mosquitoes here, and all of the mosquitoes, all of the sequence read data from mosquitoes was aligned to the AGAMP3 reference genome, and then we called SNPs from those alignments using GATK best practice. And then we did a lot of work, which I'm not going to go into, but we did a lot of work then on sample quality control and also variant filtering, site filtering, to identify the SNPs that we thought were robust and high quality. And the result of all of that analysis was that we ended up with 52.5 million SNPs across the whole of the 3 chromosomes of the Anopheles gambiae genome, which we reported. And this slide here is meant to communicate a few of the kind of statistics that we reported on those SNPs. So for example, a SNP can be biallelic or it can be multi-allelic. And so a biallelic SNP, you just find 2 different possible alleles, but a multi-allelic, you find 3 or 4 possible alleles at a given site. And 21% of the SNPs we found were multi-allelic, so they had 3 or more alleles. And when we sort of looked across the genome at how often we found SNPs, on average, we found SNPs around every 2.2 bases. So that was basically, on average, at every other position of the genome, we were finding a SNP. And so these are sort of been useful summary statistics, particularly when we compare these data with data from other species like humans, for example, because you just don't find anywhere near this density or level of nucleotide variation in comparable human genetic studies, for example. So Anopheles mosquitoes are very genetically diverse. And this plot here was meant to sort of convey, again, a little bit of information about basically the SNPs that we found and where we found the SNPs across the genome. So in this plot here, we're looking at the y-axis is variant allele density, but that really just means, on average, how often do you see SNPs when you go across the genome? So 0.5 here means every second position or base in the genome, you're finding a SNP. So over most of the genome, for example, you're seeing that the level is around about 0.5. So again, every other position you find a SNP. But there's some sort of interesting kind of fluctuations as you look across the genome. For example, so one thing we plotted here were the schematic which shows something called chromatin state. So chromosomes can exist in different kind of physical states, and towards the centromeres, which are these kind of central features of chromosomes, they exist in a different state, which is called heterochromatin. And heterochromatin is physically very different and behaves very differently from euchromatin. It's much more packed together, and that sort of physical property of the genome has an impact on the amount of diversity that you find. So one of the sort of interesting kind of pieces of biology here, when you're looking at something like genetic variation, is that the genetic variation that you see is influenced in part by the population biology, so how many mosquitoes are there, how are they interbreeding with each other, but also the kind of physical chromosome biology, genome biology can have a big impact as well. For example, we're seeing much less diversity, fewer SNPs in these regions where we see heterochromatin towards the centromeres. Although this little patch of heterochromatin here is very odd and unusual because it seems to behave in the opposite direction. We see more SNPs there than we see elsewhere. So that's an interesting exception. But another thing that comes out quite strongly here, even with this very simple analysis, is that there's certain genes here, like the glutathione transferase epsilon gene, like the cytochrome P450 CYP6P gene cluster, where you see a drop in the number of SNPs. And so those are kind of early clues that there's something interesting happening at these locations in the genome, which we'll come back to later. Okay, so just a technical point, because we did look a little bit at site filtering in the last workshop. So as part of this analysis, we did create site filters. And another name for this is genome accessibility. So sometimes we might talk about how accessible is the genome. That really means what fraction of positions in the genome pass our site filters, meaning that we think we can accurately call variation there. And basically, as a result of our analyses, we passed 141 megabases of the genome. So 141 megabase pairs passed our site filters, which amounts to 61% of the reference genome in total. So what that means is that there's 39% of the reference genome where we don't have confident ability to call variation. So there are some sort of important missing areas potentially in our data here, but we can access 61% of the genome. And particularly, actually, our accessibility is much higher within the exome. So the exome are the parts of the genome which code for proteins, code for amino acid sequences. And overall, the accessibility of the exome is around 90%. So around 90% of the protein coding variation we think we can access and call confidently. But the non-coding regions are harder to access overall, which brings the total down a little bit. And you can see also again here that some of the features of the kind of physical structure of the chromosomes have an impact here. For example, accessibility is lower in these centromeric regions of heterochromatin on all of the chromosomes. And that's generally because these heterochromatic regions have a lot of repeats in them, different types of repeat features. And so calling variation from short reads is obviously much harder in very repetitive genome regions, and that affects the accessibility, how many of those positions we pass our site filters. So these parts of the genome are basically harder to analyze and discover variation within, and that just needs to be borne in mind when you're looking at other kinds of data. In terms of how many SNPs we found, we also did another simple analysis, which is just to look in every single mosquito, how many SNPs do we find? And so this was just a simple plot where every single mosquito is like a single marker here, and we just plot the total number of variant alleles, so the total number of SNP, non-reference SNP alleles that we found in each individual mosquito. So the x-axis here is just the individual mosquitoes that we've analyzed, and the bottom line is here that overall we find somewhere between 1.7 and 2.7 million variant alleles in our mosquitoes, and there's some variation between the different cohorts that we have here. So the color here is corresponding to both the country of origin and the species assignment. So AO here is Angola Kalutsi, BF is Burkina Faso Kalutsi, GW is Guinea-Bissau, GN is Guinea, BF Burkina Faso, Cameroon, Gabon, Uganda, and Kenya. So there's some geographical variation here, but there's no systematic difference here between the species. So the Colucci mosquitoes have a similar amount of variation as the gambiae mosquitoes. And this was somewhat reassuring as well, because we were worried that perhaps the reference genome that we were using was not very representative of perhaps one species or another. But I think overall, the reference genome is more or less equally representative of both Anopheles gambiae and Anopheles colucci. And then following on from that, one of the analyses that we thought we would do is, given that we'd found a lot of SNPs, more SNPs than we were necessarily expecting to find, we then thought that we would look at the potential impact of that much variation on the ability to find viable sites in the genome where you could design a gene drive. So there's a lot of ongoing work to try and design gene drives as a potential novel mosquito control tool for vector control. These are selfish genetic elements which have the ability to propagate themselves in the mosquito population and potentially either alter the population or in a way that prevents malaria transmission or crash the population. But it does rely on finding positions in the genome which are very highly conserved, where you don't find very much variation. So we looked at the impact of the natural variation that we found in these mosquitoes on the ability to identify conserved Cas9 targets. And the bottom line was that, as you might expect, as you consider more and more individuals, so the x-axis here on these plots is the number of mosquitoes that we've looked at. As you consider more individuals, you discover more and more SNPs overall. So this first plot is just how many SNPs do we find as we look at more and more individual mosquitoes. And then this plot here is then, okay, and now how many Cas9 targets are left in the genome which are fully conserved, so there's no variation in those. And obviously that decreases substantially as you consider more and more mosquitoes, because you find more and more SNPs. And similarly, the number of genes which contain one or more of these Cas9 targets decreases a lot. So this kind of level of natural variation is potentially one of the difficulties that Cas9 based gene drives would have to overcome. But there was some sort of light here, in the sense that even though there were clearly there's clearly a lot of variation to contend with, we still did find quite a few genes which had a number of conserved Cas9 targets, including a number of sterility genes, which are good candidates for trying to design gene drives. So potentially it is still possible to design gene drives that work around this level of natural variation. But there's only limited sampling here as well, though, so that clearly further sampling of natural populations is required. Okay, so I'm now going to introduce the analyses that we did, which looked at population structure. So I'm going to sort of lift up from looking at nucleotide variation and present results from analyses that we did, which use the nucleotide variation data to analyze the population biology, the population structure of the mosquitoes that we've sampled, and also look at differentiation. So that's how genetically different are the different populations that we've sampled. So again, one of the sort of things that you have to contend with when you're working with Anopheles gambiae complex mosquitoes is the architecture of the genome, because unfortunately it's not a simple story. So population structure, as you'll all remember, is basically exploring how the mosquitoes that you've sequenced are genetically related to each other and looking for patterns. So looking for patterns where you're finding groups of mosquitoes which are more closely related to each other than they are to other groups of mosquitoes, and then relating that to various factors like the geography, so where were they sampled, and the species that they were assigned to. But unfortunately, those patterns of relatedness are not consistent across the genome. So how mosquitoes are related to each other varies a lot depending on which part of the genome you analyze, for a number of different reasons. And so to just sort of explore and try and understand that interaction between genome architecture and population structure, we first did an analysis where we looked at population structure, but independently in windows across the genome. So within, I think it was 200,000 base pair windows, in each of those windows across the genome, we performed a separate analysis of population structure. And here we computed the pairwise difference between mosquitoes, and then we turned those into some neighbor joining trees. So these trees, in these trees, every marker around the edge is a mosquito, and the branches are a representation of how closely they're related to other mosquitoes. And so you can sort of see qualitatively that just the structure of these trees looks very, very different. For example, if you look at any of the, and these are just 5 trees that are kind of representative of different patterns that we saw, and these markers kind of show where in the genome those trees came from. But if you go, for example, to near the centromere of the X chromosome, you find trees that look a bit like this. So you've got all of the Anopheles gambiae mosquitoes down one end, all of the Anopheles coluti mosquitoes way, way down the other end, and a few odd mosquitoes sort of hanging around somewhere in between. But then if you go to a different part of the genome, for example, somewhere around here on chromosome R3R, you get a tree that looks much more like this. So much less clear distinction between species and more separation by geography. So for example, the Gabon dark green mosquitoes seem to group together here. The Angola dark red mosquitoes seem to group together here, for example. So different patterns. And then you've got these large chromosomal inversions. So this is the region of the 2LA inversion. This is the region of the 2RB inversion. And those inversions, polymorphic inversions, create their own patterns of relatedness here. So these are actually the different karyotypes with the heterokaryotypes in the middle, for example. So if you just looked at the whole genome, you'd get a kind of mishmash of all of these different signals. And I'm not sure that will be very informative. So what we did is we tried to kind of partition up the genome and say, well, within these centromeres, we know that there's some kind of physical factors of the genome biology which affect relatedness and limit the ability of variation to move between individuals. So we're just going to look for a population structure analyses outside of these centromeric regions, these regions that we colored green, which all look a bit like this. So they're outside of centromeric regions, they're outside of these chromosomal inversions. So all of our population structure analysis focuses on these kind of greener regions of chromosome 3. So then just using those regions of the genome of chromosome 3, we then ran a PCA. And we're then using that PCA to explore particularly geographical factors that are affecting population structure. And the main sort of result of that is, so here are our PCA scatter plots showing from PC1 up to PC9. And so here we're plotting PC1 against PC2, PC3 against PC4. And each individual marker here represents a mosquito colored by country and species. If you just look at PC1 and PC2, the main thing that emerges is that there are kind of 5 major groupings of mosquitoes. So we have here the Angolan coluti mosquitoes, which are very, very clearly distinct. Then we have Kenyan mosquitoes, Gabon gambiae, and then we have Burkina Faso coluti and Guinea-Bissau mosquitoes reasonably close together. And then our 5th group here are basically all of the remaining gambiae. So that's gambiae from West Africa, Guinea, Burkina Faso, Cameroon, but also here gambiae from Uganda. So this sort of began to suggest for us that there are kind of some areas where there's actually very little differentiation between mosquitoes. For example, all of the Anopheles gambiae mosquitoes from a broad geographical range are not separating out very, very strongly. So that starts to suggest, for example, that there might be high levels of gene flow between certain populations. But there are also some very strong distinctions. For example, the Angolan coluti are very, very distinct from the Burkina Faso coluti. And of course, those are separated by a large distance, but they're also separated by the equatorial rainforest, which is obviously a big potential ecological feature that could affect gene flow. And similarly, the Gabon mosquitoes, so if we look at PC3 here, the Gabon gambiae mosquitoes separate out very strongly from all of the remaining gambiae mosquitoes. And here there's less geographical distance because Gabon and Cameroon are not as far separated, but they are still separated by the equatorial rainforest. So again, we began to think that, that may be a kind of barrier to gene flow, which has been kind of indicated by previous studies of population structure as well. So was something that we were looking for in particular. So we have 5 major groupings of mosquitoes, but if you do look down at the further PCs, so if you look at PC5 and PC6, you do start to see other splits coming out. For example, the Ugandan mosquitoes do split out. The Ugandan gambiae do split out from the remaining gambiae. And so from East to Central Africa, there is some differentiation among the gambiae. And then also the Guinea-Bissau mosquitoes separate out from the Burkina Faso colutsi. So there's some population structure there between those 2 groups. And to sort of confirm and cross-check those results, we also ran an admixture analysis. So this admixture analysis is an analysis which attempts to model each individual mosquito as a potential mixture of ancestries from a certain number of distinct populations. And you can vary the number of distinct populations in the model. So we tried all, and this is the value of K here, which is how many distinct populations do you have in your model. And we tried from K equals 2 to K equals 10. So what you see at, for example, K equals 5 is you see that you get the Angola colutsi. Basically, you get results that are very similar to PC1 and PC2 from the PCA. So you get Angola colutsi strongly differentiated, Gabon, gambiae, and Kenya strongly differentiated. Then the remaining gambiae are all kind of connected together as part of the same population. And then the Burkina Faso colutsi and the Guinea-Bissau are somewhat related too. But when you go down to K equals 8, you start to see some of those splits which match some of the lower PCs from the PCA analysis. For example, Uganda becomes clearly distinct and Guinea-Bissau becomes clearly distinct. And also you do start to see this potential mixture of different ancestries within Cameroon, which I think is something that still needs to be explored a little bit, because we don't, at this point, we only had data from a kind of a small region of Cameroon. So we didn't have the ability to explore population structure within Cameroon. But I think that's definitely something to explore with the more samples that we have in AG1000G phase 3. So yeah, hopefully this makes sense. Again, this is kind of a plot that's made up of lots of thin vertical bars where each vertical bar represents a mosquito, and they're colored by the ancestry that they contain. So yeah, 5 major divisions between populations, but also some weaker but still clear divisions between other populations. And we sort of ended up focusing on K equals 8 because that seemed to match closely what we found in our PCA analyses. So the next thing we looked at is population differentiation. So this is trying to quantify how genetically distant different populations of mosquitoes are. And we started with an analysis using a relatively simple statistic called FST, which just summarizes how different allele frequencies are. So you look at all the SNPs across the genome, you compare 2 populations of mosquitoes, and you look at, on average, how different are the allele frequencies between those 2 groups of mosquitoes. And so the numbers in this lower triangle here are the pairwise FST values between different populations. And this sort of brings out a few features which I've already mentioned. For example, in these West and Central African gambiae populations, there is very low, almost zero differentiation. for example, if you compare Burkina Faso gambiae to the Guinea gambiae, you have an FST of 0. So that means in practice, there's no practical difference in allele frequencies between those two groups of mosquitoes. That in turn suggests that you have high levels of gene flow, which acts to maintain similar allele frequencies between two populations. And also between our Cameroon mosquitoes, we had very low FST, 0.01 FST to the Guinea and Burkina Faso gambiae groups. So again, that suggests that there could be high levels of gene flow acting to maintain similar allele frequencies across this relatively broad geographical range. But in contrast, there are some pairs of populations where you have very high FST, so very high levels of differentiation. For example, between Burkina Faso Kulutsi and Angola Kulutsi, FST is 0.1. And so relatively speaking, that's much higher than some of these other comparisons. Interestingly, it's actually higher than between the two species. So if we compare Burkina Faso gambiae to Burkina Faso Kulutsi, FST value is 0.03. So these two Kulutsi populations are more strongly differentiated than these two different species collected from the same location. So that's a sort of interesting point of comparison where sometimes geographical separation can lead to stronger differentiation than the fact that you have two different species who have some degree of reproductive isolation. So, and again, this kind of backs up these observations of whenever we look at pairs of populations that are separated by the equatorial rainforest, we find high FST. So Burkina Faso Kulutsi versus Angola, there's relatively high FST. When we look at Gabon gambiae, again, relatively high FST. So Gabon gambiae versus Cameroon gambiae, it's not quite as high, but it's still 0.05. And also across the Rift Valley, I mean, we only have two sampling points on either side here of the Rift, the Uganda gambiae and the Kenyan mosquitoes. But the Kenyan mosquitoes have the highest value of FST to all of the other populations. So this is a relatively strongly differentiated population, including relative to the Ugandan population, which was closest. So again, that suggests that perhaps the Rift Valley system or something in that region is potentially a strong barrier to gene flow. And just to try and find a way of bringing some of those results together and presenting them in a single picture, we made this picture, but there's quite a lot going on in this picture. But basically what we did is we drew here the admixture results from k equals 8. And then we drew these pie charts, which kind of add up the different ancestry fractions for all the mosquitoes from a given location. And that really just sort of helps to reinforce where these different kind of genetically distinct groups are. So there's the Guinea gambiae, Burkina Faso gambiae, Cameroon gambiae, all of which have this kind of common coloring, suggesting they have a lot of common ancestry. And then we also kind of added in some FST, pairwise FST values, just to help kind of reinforce the fact that, so these numbers here at the top between the lines are the pairwise FST values that we just looked at. And so again, you can see between Guinea and Burkina Faso gambiae, there's 0 FST. And then between Burkina Faso and Cameroon gambiae, 0.1 FST. But then in contrast, for example, if you look at Uganda versus Kenya, you've got much higher FST, 0.2. So again, this was just meant to sort of help to bring out some of these areas where FST is very high or particularly low, and then connect that through to different geographical features. So that's as far as we got with the population structure analysis, but also in the general sort of area of population structure, we did also look at the question of species assignment, and this is actually going to be the topic of the next training workshop. So we're going to be looking at this in more detail in the next workshop. But we did know ahead of time that there were going to be some populations potentially where assigning a species was difficult. And particularly, we knew in the far west, in Guinea-Bissau, there had been a lot of reports of mosquitoes with an apparent hybrid gambiae genotype. So we knew that that was going to be interesting to investigate. And so what we did to investigate species assignment was, rather than just look at a single marker, and so the conventional molecular diagnostics for species look at a single marker. So they literally look at a single position in the genome. And that single position may not be informative. So it's possible to have a mosquito which has the marker for one species, but actually the rest of the genome looks like a different species, because we know that there is some potential to hybridize or mix between the two species. So we wanted to try and get a more kind of genome-wide view of a species ancestry. And so we did that using something called ancestry informative markers, which are SNPs, where the two different alleles are very informative of the different species. They've basically become fixed or close to fixed differences between gambiae and colucci. So we ascertained some SNPs which could be used as ancestry informative markers from a previous study, and then we painted all of our mosquitoes. So here, every row is an individual mosquito that we've sequenced in AG1000G phase 1, and it's colored according to whether its genotype is homozygous for the gambiae allele is blue, homozygous for the colucci allele is red, and then an apparent mixed genotype, gambiae colucci, is this yellow color. And then when we did have a conventional PCR-based molecular diagnostic, we just plotted that color here along the left. And the general pattern here is that for most of these populations, where the molecular diagnostic showed that it should be a gambiae mosquito, the aims in general agreed with that. So where the molecular diagnostic genotype is blue, most of the aims were in fact blue. And there's a bit of scattering here, which suggests that maybe the aims aren't perfect aims. Actually, they do kind of segregate a bit, but otherwise there's kind of strong agreement. And then in Burkina Faso and Angola, again, where we find colucci according to conventional diagnostics, most of the genome agrees with that. Although within colucci, there is one particular part of the genome here which ends up being blue rather than red. And this is related to an adaptive introgression of an insecticide resistance allele. So in both of these populations, you've got mosquitoes which have got a chunk of their genome on chromosome arm 2L, which has come from gambiae, and it's brought with it some insecticide resistance, but still the rest of their genome is mostly colucci-like. But then you get these two odd populations, so Guinea-Bissau and Kenya, where actually there isn't any sort of clear correspondence between, here is the conventional diagnostic, and then here is the picture from across the genome. And you're getting individuals that have an apparent mixture of ancestries in Guinea-Bissau, which is not that surprising. But in Kenya, we saw a somewhat kind of similar pattern, which was surprising, because in Kenya, it's not really possible or plausible that there's been any mixing or hybridization between gambiae and colucci, because colucci, the eastward limit of colucci's geographical range, is supposed to be somewhere in Central Africa. So basically, the species assignment of these two groups is somewhat uncertain and difficult to ascertain. And that's something that we are going to sort of explore in more detail. I haven't got time to sort of fully unpack that now, but we'll look at that in more detail in the next training workshop. So I'm now going to, I think, probably just touch on a few highlights from the remaining analyses, but I'm going to try and leave a bit more time for discussion, so apologies if I skip through this a little bit quickly. So after we'd done this analysis of population structure, we then wanted to look at genetic diversity and quantify genetic diversity within each of these populations, and then try and connect that back with demography, because there's a sort of very strong and intimate link between genetic diversity and the size of a population and the extent to which migration occurs between populations. So this is an idea that I just wanted to sort of plant, is that when you look at genetic diversity, you can start to begin to make inferences about demography, about the size and the history of the size of populations and potentially rates of migration. And so there's a number of different ways of quantifying genetic diversity, which we looked at. But the sort of bottom line for these various different analyses was that however we looked at it, the first thing that jumped out was always that Kenya was different. So Kenya had lower diversity, had a deficit of rare alleles, it had much, much longer range linkage disequilibrium. And so the grey Kenyan mosquitoes also had something called long runs of homozygosity. So this is regions of the genome where every individual has highly similar chromosomes. And these are all symptoms, these are all things that you would expect to see if a population has gone through a major bottleneck. So if you have a contraction in size of a population, that has a strong effect on the genetic diversity of the population, which you would expect to look somewhat like this in all cases. And so the interesting follow-on question then is what caused, or what could have caused, such a population crash? And is it anything to do with vector control, or is it actually something related to something more natural, such as variation in climate? And we had a go at trying to answer that question, but some of these methods for inferring population size history from genetic data are somewhat in their infancy. So there's still, I think, a good deal of uncertainty about these inferences. But we ran a program which basically can infer, going back over time, over the number of generations. So this is the present time. So going back over time, at what point, what was the size of the population at different points in time? And so these lines are our inference of the size of the Kenyan population going back over time. And we repeated this analysis for 2 different chromosome arms separately, just to see how well they agreed with each other. And the sort of outcome of this was suggesting that the decline in these populations had begun somewhere around 200 generations before the time of sampling. And so if you have something like 10 generations a year, this would put this decline well before the scale up of vector control interventions. So it suggested that this population had been declining before bed nets were introduced. But that is a very sort of speculative conclusion. So I think there's a lot more that you would want to do to actually kind of investigate that if you wanted a firm conclusion. I'm going to skip over this, which is looking more at ancient population size history, but very happy to answer questions on it if anyone's interested. Now, I'm just going to briefly then touch on the final analyses that we did, which were looking at selection and adaptive gene flow, particularly because we wanted to learn something about insecticide resistance. And so there were 2 parts to this analysis. The first thing we did was run some genome-wide selection scans. So these are analyses which look across the whole genome, and they look for genetic signals that there's been recent positive selection at a particular place in the genome. And we'll run these analyses for ourselves in a couple of workshops' time. But basically what you're looking for here is you're looking for peaks. And every time you see a peak, like this one here, for example, that's a signal that some gene or some variant at this position in the genome has been strongly positively selected for, which is exactly what you'd expect if there's, for example, an insecticide resistance mutation at this locus. And so this begins to give us a genome-wide view of which genes are being naturally selected for in the recent past of these populations, which helps us understand a bit more about which genes are kind of playing an important role in the adaptive response to vector control. And out of this analysis, we find some of the usual suspects, so some known insecticide resistance genes, like the voltage-gated sodium channel gene, like some cytochrome P450 genes. But there are also some peaks genes which have not previously been associated with insecticide resistance. So there's a peak here, for example, which I think is an interesting peak to follow up. And there's a peak here, for example, again, which is an interesting peak to follow up. So there's here the kind of potential to discover new genes and new variants that we didn't previously know were being involved in insecticide resistance adaptation. And the last thing was just to mention that, so we did a first pass analysis of variation, particularly within the voltage-gated sodium channel gene. And we've already looked at this in more detail because Chris covered actually a later analysis that we did of this gene, which went into a lot more detail. But I thought I'd mention just this part of it, because what we found when we looked at this gene, and what we particularly did was we looked for sharing of haplotypes that carry resistance alleles. And we found a number of haplotypes or groups of haplotypes which were almost genetically identical, but where the haplotypes came from mosquitoes from a number of different locations. And when you find this degree of genetic similarity, that's an indication of very recent gene flow. And so what we could start to do from this type of analysis is infer cases where insecticide resistance has spread and has spread to a number of different populations. And this surprised us because we found a number of examples of the spread of resistance, which contradicted our kind of results of population structure analysis, because we found sharing of insecticide resistance haplotypes between populations that were otherwise very strongly differentiated. For example, there's a haplotype here, S3, which you find both in Kenya and in Uganda. So despite the fact that these two populations seem strongly differentiated, which suggests low rates of gene flow, still there must be some gene flow, and it must be enough so that when you get a very strongly positive selected variant, it can spread to different locations, either on either side of the rift, or in these cases on different sides of the equatorial rainforest. So what did we conclude from all of this analysis at the time? Well, we tried to draw this back to challenges in vector control, and we tried to make the case that yes, we need new insecticides, and yes, we need new vector control tools and strategies, but we also need better tools for surveillance, for gathering intelligence, to help us plan interventions, and to try and get ahead of mosquito evolution, basically. And so we propose that what we sort of have here is a proof of concept that genomic data could become a surveillance tool and could be collected as a part of vector control interventions, so that we can ask questions like, what is causing insecticide resistance to emerge or spread, or what causes a mosquito population to crash, and ultimately start to think about every intervention as an experiment, and gather data that allows us to analyze its impact on the mosquito population, and thereby try and improve our interventions going forwards. So I will leave it there, but thank you so much for listening, and look forward to questions.
So we don't have any questions in the chat yet. Anyone who has questions, please drop them in. I guess for now, I have a question. When you've added other mosquito samples and done some other analyses, has there been any striking sort of disagreement with the current results from this study?
No, I don't think anything has contradicted anything that we've seen so far. I think what it has done is help us to sort of just gain a little bit more. Generally it sort of corroborated results, and then just given us a bit more kind of detail around where we see, for example, strong population structure, or where we see uncertain species assignments. So for example, when we added in mosquitoes from the Gambia in the far west near to Guinea-Bissau, we found mosquitoes with apparent mixed species ancestry. And so clearly what we were seeing in the Gambia was not a one-off. It's something that you see in other populations nearby. Similarly, in the far east of Africa and coastal parts of East Africa, what we saw in Kenya, we're also seeing in some regions of Tanzania as well in terms of uncertain species assignment. So, so those, I think, have sort of helped to support the idea that there are some interesting and unusual phenomena which do need to be investigated. And then in terms of population structure, particularly around the equatorial rainforests, we're starting to try and, because although we're seeing strong differentiation across the rainforest, we only have a few, at this point, we only had a couple of data points on either side. So there's big questions, I think, there about actually where are the boundaries between these genetically distinct populations, and where do you see gene flow versus where don't you? So starting to fill in some of those questions is beginning to happen with the further data that we have.
So now we have some questions rolling in from Mugenzi. Thanks, Alistair. Why use genetic crosses to evaluate data quality as part one? And then we have some questions about PCA and admixture.
So the genetic crosses, with genetic crosses, because you know who the parents are and you know who the offspring are, you can use your expectation about the fact that if the offspring can only get their genotype from one of the two parents, or for a combination of the two parents. So you have inheritance as a way of cross-checking the consistency of your data. And so you can look at every single SNP in your crosses and you can say, the genotypes that we've called, are they consistent with inheritance of alleles from parent to offspring, or are they not consistent? And if they're not consistent, that's a big clue that your data are of lower quality. And so the name for that is Mendelian error. And we look at Mendelian error as an indicator of poor quality data. And then we calibrate all of our site filters using Mendelian error. So we basically aim to filter out SNPs which have higher rates of Mendelian error. So the question about PCA was looking at the PCA plots, which is the most informative using PC1 versus PC2?
And maybe generally following up on some questions from the last workshop, like how does one read a PCA?
Yeah. Well, if I perhaps skip back to that. So the simplest way of reading these is basically just to look for where you have clusters or groups of individuals which are clearly separate from other groups of individuals. And so in, for example, PC1 versus PC2, you can see that there's a number of very clearly distinct groups of individual mosquitoes. And that tells you that the individuals from Angola, for example, are more closely related to each other than they are to any of these other groups of mosquitoes. You can cross-check that against the variance explained. And you can see, for example, that PC1 and PC2 explain quite a lot of variance relative to the tail of principal components. So it suggests that PC1 and PC2 are very informative. You're seeing clear structure here, clear separation between different groups of mosquitoes, and higher variance explained. And PC1 and PC2 are always going to pull out the strongest separations in your data set. So this is telling you where you have the strongest differentiation between different populations. But I think all the way down to PC6 is informative here, because again, you're seeing kind of clear splits between different groups of mosquitoes that does correspond very clearly with geography. So it's very, very plausible. You're seeing Uganda split out here, you're seeing Guinea-Bissau split out here, for example. So I think down to PC6 is all very informative about genetically distinct groups of mosquitoes. PC7 to 10 are a bit more interesting, because you're getting some spread, but only within the mosquitoes from Cameroon. So I think there's an interesting question about what's driving this, and is this real structure within the Cameroonian mosquitoes, or is this some artifact of PCA, or there's other things that could be happening. For example, if you sample some partially related individuals, that can also come out on PCA. So we looked at up to PC6 as giving us our main information about population structure. Hopefully that helps.
So we just have a few minutes left, and there's a few questions about what are the key factors driving genetic diversity, environmental factors or epigenetics, and in places where you see multiple populations of Anopheles gambiae and Kalutzii, do you see evidence of gene flow between them?
Yeah.
So sort of general gene flow and differentiation questions.
Yeah, so we, in terms of what determines genetic diversity, the key factors are population size, history, and also migration. And the reason why those are key factors is because the size of the population determines genetic drift, determines how fast diversity is removed from a population. So large populations can sustain a lot of variation for a long period of time. So you accumulate a lot of variation, diversity within a population. So the bottom line seemed to be that all of these populations to the north of the equatorial rainforest had high diversity, and that was consistent with very large effective population size, but also actually was consistent with population expansion too. So it seemed like all populations to the north of the rainforest had gone through some sort of quite significant historical expansion in population size. And there's a very interesting question there, historical question about what might have driven that, which we had a bit of a go of discussing, but we don't have a clear answer for. So hopefully that, but the interesting thing was that the populations south of the equatorial rainforest did not share those same patterns of diversity, did not appear to share the same degree of population size or population size history. So whatever happened north of the equatorial rainforest did not seem to happen in these other populations. So that's another interesting question. Why do you see that difference? And yes, in terms of gene flow, clearly there are multiple examples of gene flow between species, despite differentiation, and between geographically distinct populations. And the examples of insecticide resistance alleles just give us that kind of clear demonstration that populations are clearly connected. Almost all of the populations that we've sampled here, despite strong structure, are connected by some degree of gene flow. So resistance alleles have the potential to move potentially large distances, which I think is something to bear in mind. And,
so I think we'll have to wrap it up there, but thank you very much, Alistair, and thank you everyone for joining us. We'll see you next time, and if you have follow-up questions, you can always drop them in our Slack channel and someone can get back to you. So thanks, bye.
Thanks all.